Today I'm going to be talking about a research proposal on phytoplankton diversity, specifically within various marine protected areas. I will go through a brief introduction, methods, some advancements to general knowledge, and broader impacts. Primary producers are the base of our food webs, and they have the ability to produce organic compounds from chemical or light energy that they receive. The primary producers I will be discussing are phytoplankton, who receive their energy from the sun. Here I've listed a few different types of phytoplankton, including diatoms, dinoflagellates, cyanobacteria, and green algae. Biodiversity is an important factor in all ecosystems. Environments are more productive with higher biodiversity and become more adaptable to changes when species richness is high. The ocean plays a major role in the regulation of the climate. With higher biodiversity comes a more productive marine ecosystem that is better at moderating the climate. Marine protected areas, or MPAs, are defined by NOAA as a defined region designated and managed for the long-term conservation of marine resources, ecosystem services, or cultural heritage. NEOLI is an acronym created for the characteristics that contribute to the success of an MPA. NEOLI stands for no take, enforced, old, large, and isolated. The question at hand in this research proposal is, do marine protected areas that exhibit higher success rates tend to have higher phytoplankton diversity? Given the known importance of both biodiversity within an ecosystem and primary productivity for the food web, we expect areas of healthier composition to include higher diversity of phytoplankton. If we can accept the rationale that MPAs with more NEOLI conditions are more successful, and success is reflected by a healthier ecosystem, we would hypothesize that MPAs with higher success would indeed tend to have higher phytoplankton diversity. I chose 4 MPAs from each level of success to sample. I would then use Niskin bottles attached to rosettes to collect 50 to 100 milliliter samples of phytoplankton to be stored in Lugol solution. These samples would be analyzed and counted under a microscope, identifying the species when possible. This data would be portrayed graphically, comparing the samples both studied within MPA sites and between MPA sites. This project would advance our knowledge on how MPAs help strengthen our ocean's biodiversity at all levels. It has the potential to inform scientists on how to better implement MPAs to improve ecosystem function. This research can show that phytoplankton are important beyond their blooms. They can be ecosystem health indicators that can inform decision-making. Following this study, public outreach could be conducted geared towards our upcoming generations. It could also provide education to the public, spreading the message that we must continue to protect our ocean, even at the smallest of scales. Thank you for listening to my proposal, and here are my resources.